So we will take up now the amino acids and peptides. That's chapter 3. Okay. Okay. So amino acids, as you can see there are 300 or more which occur in nature. But we only need 20 of those amino acids to be used by our cells to form proteins. So you can see this at table 37-1, where the amino acids have different codons. Now the codon is your 3-letter code, which is found in your messenger RNA, and it is translated by your ribosome in order to be formed into proteins. So they are stringed together, one amino acid to another through a peptide linkage or a peptide bond. Now, this codon, as I mentioned, is a 3-letter code. And of course, you know what those letters are. The letters are the first letter of your pyrimidines and your purines. And your purines, you have adenine, guanine. Your pyrimidines, you have thymine, uracil, and what's the other one? Huh?
Cytosine.
Cytosine. Very good. Okay. So at least you have an idea already. So I'm just asking you so I can find to be able to know whether you have an idea of what we are talking about. Okay. Now this is the encoded in what you call your genetic code. Although this genetic code can accommodate more than 20 amino acids, if there are other amino acids that can be utilized aside from these 20 in the formation of our human protein. But it's not, it's only that there are only 20 amino acids. So this 3-letter code becomes redundant, meaning to say that one amino acid may have several codons. Okay? So you have there, let's look at our amino acids. Okay? So they are grouped together. First is with aliphatic side chain. So you have fire, glycine, alanine, valine, leucine, and isoleucine. All right, your glycine is the symbol for glycine. You have two, a single letter abbreviation or a 3-letter abbreviation. So if a 3-letter abbreviation for glycine is GLY, and single letter is represented by G. And you have there the structural formula. You have the amino group and the carboxyl group. The amino group and the carboxyl group. And this one here is supposed to have carbons that are attached here. But your glycine does not have additional carbons. So this is supposed to be what we call here the R group, as shown here. Look at that. There's a methyl group here. No? Or this one. You have a very big R group there. This one also, a very big R group. Okay. So glycine has no R group. Now, if you still remember your kindergarten organic chemistry, now it's this carbon here, or this one, which we call the alpha carbon. Actually, this alpha carbon is carbon 2 of your organic acid, remember? This is carbon 1, this is carbon 2, that's carbon 3. But if you use your Greek letter, this is alpha carbon and this is beta carbon. No, you remember that? Okay, now going back to this. You will notice that this carbon here, the alpha carbon, has two hydrogens attached. No? While if you look at this carbon here, all the attachment of your carbon are different from each other. One is hydrogen here, this methyl group, this is the ammonium group, and this is the carboxyl group. What do you call that? When a carbon has four different atoms or group of atoms attached to it, what do you call that carbon?
Chiral.
What's that again?
Chiral carbon.
Yeah, all right. That's correct. No? So, your glycine, no, because these two are the same, hydrogen. So this is the only amino acid in which you do not have a chiral or chiral, no, carbon. All the amino acids, the alpha carbon is chiral or chiral. Okay? All right. So you have aliphatic side chains. And since they are aliphatic here, the side chain here or the R group is not soluble in water. So which means they are not soluble in water and you call them hydrophobic amino acids. They do not like the water. Okay. Then we have a hydroxyl containing amino acid. We have serine, threonine, and tyrosine. Okay. Serine here, hydroxyl group. You have an OH here on the R group, an OH here. Now for tyrosine, we will look at this when we go to the aromatic group. No? Tyrosine contains a hydroxyl in its aromatic group. Okay. Then we have sulfur containing. We have only two, cysteine and methionine. So you have here sulfur, ethyl group, and methyl thio. Okay. For your methionine. Okay. Amino acids containing acid group or their amides. Now, aspartic acid and glutamic acid, they contain carboxyl group here. Carboxyl group. Okay. So there are two carboxyl groups already here. So that means they tend to be acidic. So you have aspartic acid and glutamic acid. However, this carboxyl group of your aspartic acid in your beta carbon, no, carboxyl group of your beta carbon, alpha, beta, Then you have a carboxyl group. So this carboxyl group attached to the beta carbon can react with an ammonium group. And you have what we call an amide. You see this? Okay. One of the hydrogen or one of the oxygen is removed, and one of the ammonia hydrogen reacts with. So it forms now, you have ammonium group, you have now water as a result. And so you have this amide. And the amide of aspartic acid is asparagine. And so with your glutamic acid. Anong doon naman i ang acid? Oh, hindi lang. So this is alpha carbon, beta carbon, gamma carbon. To the gamma carbon of glutamic acid, you have a carboxyl group. And that carboxyl group reacts with an ammonium group. And that makes it an amide. Uy, lukso man. Hindi makita. Resobos. Here. That is an amide group here. Similar to that one here. And so the amide of glutamic acid is glutamine. Okay? All right. Then we have basic groups. Oh, come. So we have here arginine. And what contributes to the arginine becoming basic is because of these two amino groups here. Arginine. Then we have lysine. Also, this is basic here. Then we have histidine. Okay, the imidazole group here is basic. Okay. So histidine for containing aromatic rings. You have seen that histidine. And then phenylalanine. You have phenyl group. And then you have tyrosine, a phenyl group with an OH. So tyrosine belongs to aromatic as well as hydroxylated amino acid. And then of course tryptophan. No, you have a big aromatic group here. Very big aromatic group. Okay. And then the last is an amino acid. Now, this amino acid is made up of 1, 2, 3, 4, 5 carbon atoms. Now, you have an amino acid that we mentioned already that has 5 carbon atoms. Look at this. 1, 2, 3, 4, 5. Okay? Glutamic acid. Now, this one here, this carboxyl group, reacts with the amino group here. So they will form a ring. And that is now proline. There you are. Okay. So that's carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. It's supposed to be a carboxyl group. And then we have a amino group here. The carboxyl group and the amino group combine together to form this ring. Okay? You know that already. It's just a review. Now, so because of the R group, which also gives property, we mentioned already R groups that are soluble in water. There you are. Okay. And then you have your hydrophobic, in which you have aliphatic side chain. You have this one here, what do you have here? You have an aromatic ring. You have an aromatic ring. You have an aromatic ring. Okay. Now, this one, selenocysteine, is now considered as the 21st alpha L amino acid. So we have here L amino acid. Now going back to your organic chemistry, what does L means? Capital letter L. Hmm? Or capital letter. Well, how was that again? What is that?
Laboratory.
No. L is not because of rotation. That does not mean the capital letter L does not mean laboratory. Okay? That is a configuration or your L or your D. Yeah? Meaning its position in space. While your rotation is represented by a plus sign and a negative sign. Dextrorotatory and levorotatory. Dextrorotatory is plus, levorotatory is negative. Okay? Now, when we take up our carbohydrates, especially your glucose molecule, you can see now this capital L, capital D. Now, we'll go into that later on. All right, so we have here the 21st protein amino acid. Now, although this is not, what you call uh, coded in the genetic code, no, this amino acid is coded in the tRNA. Actually, it is the tRNA, no, which is the transfer RNA called the tRNA-seq, no, for selenium, which utilizes the codon, anticodon UGA, which is a signal for stop. Stop, meaning no more translation. However, the synthetic apparatus for protein, I can identify a selenocysteine specific UGA codon by the presence of an accompanying stem loop structure. The selenocysteine can be inserted into a protein. So this is a very unique amino acid, selenocysteine. Okay. So this is your cysteine, and this one is with selenium, selenocysteine. This is not uh, post-translational. Post-translational meaning after the translation of the coded message in the messenger RNA, then the amino acid that is already being linked to with other amino acids through a peptide linkage, this is being modified. Example, the proline and lysine that is found in collagen, that's the most abundant protein in the body, which is found in collagen, is hydroxylated. But when they are not yet inside the protein molecule or linked to other amino acids, there is no hydroxyl group. But only when they are already part of the protein, which you call them a residue. The amino acids that are found in the protein are called residue. No? So for your proline and your lysine, they are called prolyl lysyl when they are already part of the protein or any amino acids. So you have glycyl, alanine, methionine. Okay. So when they are part, that's what we call amino acid residue. So when they are already a part of the protein, amino acids known as hydroxylases comes around and add a hydroxyl either to proline or to your lysine. So you have now hydroxylated proline and hydroxylated lysine, and they are called hydroxyproline and hydroxylysine. And these two proteins are abundant in your collagen. Okay. So here we are, post-translational modification, confer additional properties. Confer additional properties because the R group now has an OH. And you know OH is water soluble. So that would diminish its being a hydrophobic group because of the presence of an OH. Okay? Oh. Hydroxyproline on the left side, here, hydroxyproline, you have the OH, and here is your lysine and the OH, hydroxylysine. So this is another post-translational modification. Your glutamic acid, now look at this, has another carboxyl group that is added. This is the alpha carbon, beta, gamma. And then you have carboxyl group there, and then another carboxyl group that is added. So this is now known as gamma carboxyglutamic acid. Now this amino acid is very important in blood clotting. Okay? Oh. extraterrestrial amino acids have been detected in meteorites. Those who are interested in aliens, if there are life other than ours in this planet Earth, meteorites that comes down to Earth, they were able to find amino acids. And you know that amino acids is very, very important in life formation. So this particular meteorite, 20,000 metric ton, very, very big. It's a good thing it landed in Siberia where there are no people, just plenty of ice. No? This is in Siberia. And then when they uh, examine the meteorite, they contain this particular amino acids, alanine, aspartate, glutamate, glycine, isoleucine, leucine, phenylalanine, serine, threonine, tyrosine, and valine. Okay, so this can can be an evidence that there is extraterrestrial life. Now we do not know if this life forms in other planets are like us, or they can just only be bacterial in form or viral in form. Okay. Now, aside from the function of the amino acid as part of the protein molecule, certain amino acids have specialized, can become specialized products. Like for example, tyrosine. Tyrosine here is the precursor of thyroid hormone. No, your T3 and T4. Okay? And then tyrosine and phenylalanine is metabolized into epinephrine, no? And norepinephrine. And DOPA, dihydroxyphenylalanine. Okay. Now, your glutamic acid, aside from being a part of a protein molecule, is both a neurotransmitter, glutamate. You're familiar with monosodium glutamate, di ba? I think you eat that every day.
Bitsin.
Bitsin, okay? And what is glutamic acid? A neurotransmitter. It can affect your nerves. Okay, and also it can be transformed into GABA or gamma aminobutyric acid. Now we have amino acids ornithine and citrulline in our body, which do not become a part of proteins, but are intermediates in the urea cycle. No? And homocysteine, homoserine, and glutamate gamma semialdehyde are intermediates of protein amino acids. And there are other special products of amino acids. We will be going into that. You know? Like for example, uh, glutamic acid, cysteine, no, and glycine. Those are the three amino acids uh, that forms your glutathione. No? How many are using glutathione? Now, glutathione is not an essential because it is not an essential nutrient because your body can form it. Your body contains glutamic acid, cysteine, and glycine, and they can be synthesized into glutathione, and they are not what we call essential amino acids. So they are common. Now, when you take supplements of glutathione, it is digested back into glutamic acid, cysteine, and glycine, in which your body already has those amino acids. So what's the use of fortifying yourself with glutathione? Diba? Well, all right. Certain plant amino acids can affect human health, especially this particular plant, lateros. This is a legume, just like your string beans. No? Oh, okay. This is implicated in a disorder known as neural no, neurolatherism, which is a neurologic disorder which brings about paralysis of the legs. Latherism occurs during famine when the lateroseeds is used as food. So this brings about paralysis. Okay. The seeds, okay, here we are. That's lateros consumed during famines and contain this particular amino acid. And this can bring about, as we said, paralysis of the legs. Other toxic amino acids, we have homoarginine, which is the one that causes neurolatherism. Then we have this oxalyl diaminopropionic acid or beta ODAP. It's a neurotoxin, which is also implicated in neurolatherism, paralysis of the legs. And there's another one, the BAPN. This is osteolaterogen. The bones are being affected. Another one is diaminobutyric acid, which is an inhibitor in the urea cycle. So your urea cycle will not be working and ammonia intoxication will be formed. And then we have this. So which produces neurodegenerative diseases. No? Well, anyway, these are not common in our food chain. So there's not much of cases reported because of the toxicity brought about by this particular uh, amino acids. Now, we mentioned earlier that the amino acids that are utilized in the formation of protein belongs to the L variety. But there are D amino acids that occurs naturally, uh, but they are not utilized by our body, like D-serine, D-aspartate, D-alanine, D-glutamate. The amino acids are found in antibiotics produced by bacteria, fungi. No? Then we have D-methionine, D-tyrosine, D-leucine, D-tryptophan to trigger biofilm disassembly and Vibrio cholera. So many incorporates D-leucine, D-methionine into the peptide component of its peptidoglycan layer, or what you call the bacteria envelope. Okay. Functional groups of amino acids. Okay, as you notice, the carboxyl group is negatively charged and the amino group is positively charged. All right. So let's look at this. Here is aspartic acid. By changing the pH where you put your aspartic acid will have different charges. Okay, let's start from here. Where you have your aspartic acid in strong acid below pH 1, and you will notice that you have your amino group that is protonated and becomes positively charged. So the net charge here is positive 1. The carboxyl group is also protonated. The hydrogen is there, the protein is there, the proton is there. You see? So at a very low pH, aspartic acid shows a positive 1 net charge. Now, if you put it a little with a little what you call this, you change it and the pH becomes 3, meaning you put some base here so that it will become pH 3. You will notice that the charge is 0. Look at this. Why? Because the OH here loses its hydrogen. Lost. And the amino group has a positive charge. So One positive, one negative equals zero. Zero charge because this cancels out. Okay? Now, if you put the pH a little bit higher from pH 3, the carboxyl group here loses its proton and becomes negatively charged. And this one here still becomes negative charge. But the amino group is still positive. So two negatives and one positive becomes net charge of negative 1. Then you increase the pH to pH 11, which is now alkaline. Okay, you will notice now that you have a negative charge here, same as this one. Oh, anyway. Same as this one here, same as this one, all negative. And then the amino group here, oh, then the amino group here loses its positivity. So there is no positive charge there. And your carboxyl group still has a negative charge. So two negative charges and zero charge in the amino group will have two negative charges. This is what we call the protonic equilibria, where the amino acid changes its charges in different protonic equilibria, where the amino acid changes its charges depending on the pH of the medium. Huh? Okay? Now, what is the physiologic pH? What is the normal pH?
7.
7.4. Okay? 7.4. It is slightly alkaline. Slightly alkaline because it is above 7. 7.4. Okay? Now, at that pH, your species of amino acid is this one. It has two charges, positive charge and negative charge. And you call that dipolar ion. Diba? Dipolar ion. Or term sweeter ions. Sweeter is a German word for two. So that means positive and negative charge. Now, this species cannot exist in your body at a 7.4 pH. Okay? But sometimes when we do reactions, we use this formula. Diba? Okay. Peak values express the strength of the weak acid. What do you mean by pK? What do you mean by pH?
Power of hydrogen.
Negative logarithm of the hydrogen ion concentration, di ba? Now what about POH? Negative logarithm of hydroxyl ions. Okay. What about pK? It's the negative logarithm of K. And what is K?
Dissociation constant.
The dissociation constant.
Potassium.
The dissociation potassium K. No, pK. All right. Dissociation constant are the acid or a base, or for that matter, water. The pK of water, or the negative logarithm of the dissociation constant of water when we ask for the pK of water. Now, what is isoelectric pH or PI? There you are, oh, an amino acid bears no net charge. Huh? Okay, going back to this. Where is the PI of aspartic acid? A, B, C, D.
B.
Oh, B. Because it's zero charge. Yes, it's zero charge. No charge at all. Because positive cancels the negative. So no charge. Zero charge. So that's the PI. No? And you get the PI of aspartic acid by adding these two, no? The pK here and the pK here divided by 2. And that is the pK of aspartic acid. This is the negative logarithm dissociation constant at this pH of aspartic acid. This one here is the pK of aspartic acid at this pH. Huh? Did you get it? Okay. Don't worry, I'll not be asking you about this. I just want you to know what this is.
Yay.
Huh? Ayay.
Mauna diha, Doc.
Mao na. Mao na. Di tamutanan ninyo, anak.
Salamat.
Okay, pK values vary with the environment. And what is the environment here?
The cell membrane.
Yes. What is the environment where you have this amino acid? Whether environment is acidic or basic. So it varies. As you have seen the protonic equilibria of aspartic acid. So it changes. No? Like the environment in your gastrointestinal tract, in the stomach, it is too acidic. Correct?
Yes.
And when you go into the small intestine, it is so alkaline. Correct? Oh, so you have two environments there. Okay? All right. The solubility of amino acid reflects their ionic character. Yes, we have already mentioned this. We have classified our amino acids into hydrophilic and hydrophobic.
Phobic.
Okay. All right. It says here, soluble in polar solvents. Sama ni. Mulukso lagi. Okay, the polar solvents such as water and ethanol, no, are those what we call as hydrophilic. Hydro, water, philia, love. So you are water lover, no? Okay, so when we say hydrophobic, phobia, no, does not want the water. So hydrophobic or non-polar. Okay, so we have here polar solvents such as water. And then the non-polar such as benzene, hexane, and ether. So these are the solvents for hydrophobic. Amino acids do not absorb visible light, and those are colorless. However, tyrosine, phenylalanine, and tryptophan, because of its aromatic group, absorb high wavelength at 250 to 290 nanometers ultraviolet light. Okay, so we have it here. So it says 250 to 280. So we have here 250. Phenylalanine is already starting to rise at a peak of 260. Okay, and then you have your tyrosine rising and at a peak at 280. While tryptophan, you have here again 250 up to, this is more than 280. No, if you drop it down, and that is 290. So this the absorption. No? Okay. The R group determines the properties of amino acids. Okay. You know the R group there. We made a generalized classification of the R group, hydrophilic and hydrophobic. But some R group has other groups that are added to it. Then that can also give you another property, right? Okay. Okay, so it gives you characteristic chemical reactions. Carbolic acid groups are for the formation of esters, amides, and acid anhydrides. The amino group, you have acylation, amidation, esterification. And for OH, and the thiol group is its group, oxidation and esterification. Since glycine, the smallest, and does not have an R group, can be accommodated in places inaccessible to other amino acid. It is often found, excuse me, excuse me po, peptide bends. So when a polypeptide bends, or peptide bends, you will be sure that you have a glycine molecule there. The hydrophobic R groups of alanine, valine, leucine, isoleucine, and aromatic R groups of phenylalanine, tyrosine, and tryptophan are found in the interior of cytosolic proteins. Cytosolic proteins meaning proteins inside the cell. Okay? Now, inside the cell is an aqueous solution. It's a water solution. So, we know that these amino acids are hydrophobic and they do not like the water. So, what do? They group together. No? Just like what I've said last time, birds of the same feathers are, oh, birds of the same feathers flock together. No? Or some says, birds of the same feathers are still birds. So, they do not like the water. So they hug each other so that the protein will have contortions. No? So as much as possible that they hug each other so close to each other that no water molecule will be between them. So that's the characteristic. So you find this amino acid of a particular protein inside, no, inside the protein, and the amino acids that are hydrophilic are outside. Okay. Okay. The charged R group, like the basic and acidic, stabilize specific protein formation via ionic interaction, or what we call salt bridges. Ionic interactions. These interactions also function in the charge relay. Charge relay. We will be looking into this. Charge relay during enzymatic catalysis or activity of the enzyme, no? And the electron transport chain in the respiring mitochondria. Okay. Amino acid sequence. Amino acids that are stringed together through peptide linkages, okay, is what we call the primary structure of the protein. So that if you have just a peptide with amino acids that are being stringed together by peptide linkages, this one, we call it a primary structure. Now, each of these amino acids that are already stringed together by your peptide linkages are called residue. So this is the residue of alanine, this is the residue of cysteine, this is the residue of valine. All right. Now, once you have this string together, okay, the residue mentioned will end in YL. And the last residue is not changed. So you may have several here, no? Like glycel, methionine, serine, alanine, cysteine, valine. You don't call it valel because this is the end. This is the last. So the number and order of the amino acid residue in a polypeptide constitute what we call primary structure. The structure of a protein made up of primary, then you have secondary, then you have tertiary and quaternary structure. So look at this, the example here. If you have here lysine, leucine, tyrosine, glutamine, so it is called lysyl, leucyl, tyrosyl, glutamine. So the INA ending on the carboxy terminal residue, and that is glutamine, remains, no? Okay, so here is the use of those 3-letter abbreviation. Glutamate or glutamic acid, alanine, lysine, glycine, tyrosine, alanine. And they are represented by the single letter. If you go back to that table of amino acids, you have there the 3-letter symbol and the single letter symbol. Okay. All right, the peptide structures are easy to draw. Okay. They usually draw it this way, no? That's the alpha carbon. No? This is the carboxyl carbon. Then you have your amino group. You have alpha carbon. Then they have carboxyl, then amino, then carboxyl, and so that's the one there. Now, see? That nitrogen is this nitrogen here. This is the alpha carbon. Alpha carbon there. Okay. That carbon is this one. That's the carboxyl carbon. And the amino carbon, the amino group. And the CO in its forms what you call your peptide linkage. Okay. Now here is another CO in its linkage. Okay. Some peptides contain unusual amino acids. Unusual amino acids. In mammals, the peptide hormones typically contain only 20 codon specified alpha amino acid linked by standard peptide bonds. Other peptides may, however, contain non-protein amino acids derived from the metabolism of the amino acids. For example, glutamate, the glutamate of glutathione, no, and the reduction of your disulfide bonds cysteine by a non-peptide bond. Okay, the amino terminal glutamate of thyrotropin releasing hormone is cyclized to form what we call pyroglutamic acid. And the carboxyl group of the carboxyl terminal of a proline residue is amidated. So an amide is added to it. Okay, so these are uh, already amino acids that is modified through post-translation. Okay, so you have here your glutamine. So many look so many. Now this one is a gamma glutamyl. Now there is a gamma carboxyl group. This carboxyl group here. This is the gamma carbon. That is the alpha, beta, gamma carbon. This is the carboxyl group of your. So you have gamma, glutamyl, cysteine, glycine. So you have here glutamate, and this one your cysteine, have an SH group there, and then this is your glycine, and this is your glutathione. Okay. The peptide bond, okay, this is the peptide bond. This is the carboxyl group of one amino acid. This is the amino group of the other amino acid. There is only one bond, but it has a character of a double bond. Although it is shown as one bar here, no, it has a character of a double bond, meaning to say that this carbon and this nitrogen cannot be rotated. Cannot be rotated because it has a double bond character. It is not like this one, nitrogen to hydrogen. You can rotate this. Okay, so you can rotate this. But this one, you cannot rotate this because it has a double bond character. Although it is not shown here, that is still a single bond, but its character is a double bond. It cannot be rotated. Okay? So this is how this is being. So from one alpha carbon to another. So one alpha carbon, this is alpha carbon, to another alpha carbon. The length here is 0.36 nanometer. Now, although this is not important, I'm just only asking this because when I took my board exam, I was not able to answer this. What is what is the link between two alpha carbons in a polypeptide chain? Two adjacent alpha carbons. No, that's 36 nanometer or 3.6 angstrom. Don't worry, I'll not ask you that. I just mention it in passing. That is not quite important in medicine. We have to measure the alpha carbons of the patient, the distance of the alpha carbon. Well, just helps in our understanding now, but that is not really important in medicine. Okay, so as you can see it here, now this rings here, what you see, this is, you can turn it around. You can turn it around. But this one, you cannot. This is a double bond character, like that one, a double bond character. Now here, you can turn it around, you can turn it around, you can turn it around, you can turn it around. But this one cannot be carbon to nitrogen. Although it is just a single bond. No? Okay. Non-covalent forces constrain peptide conformations. Okay, the folding of the protein. First, it's you have your the hydrogen bonding, the hydrophobic bonding interactions. These are forces that folds the protein. Now, we know what is a hydrophobic bonding brought about by hydrophobic amino acids. They group together. Even if they are located, the peptide length here, let's say, there are 50 amino acids in that peptide. And amino acid leucine is number 2 in the sequence. And you have another one, amino acid isoleucine, number 38. They are far away from each other in the chain. But they come together because both of them are hydrophobic. So they are attracted to each other. When you place that that peptide or polypeptide in water, so they do not like the water, so they are attracted to each other. So they form a way in which no water molecule will be between them. So the polypeptide bends as a result of that. And if there are so many bends like that, no? So hydrophobic. Another one is your hydrogen bonding brought about by your OH and the carboxyl group. No? The OH, we mentioned that the proline and lysine residue in the protein collagen, these OHs of the proline and lysine are being used to form hydrogen bonds. So much so that there is so many bondings of hydrogen bond in the polypeptide chain, in the triple helix of the collagen, and makes the collagen strong. Okay? Without the hydrogen bonding, the collagen molecule is very, very weak, as shown in what is the condition brought about by the absence of vitamin C. What do you call that condition? Allah, kalimtan. Scurvy.
Curvy.
Scurvy. No? Scurvy. Now, we do not have frank scurvy at present, where if you only hold the hand of a person with an acute scurvy, that skin will be sloughed off. You can see how weak is the skin. And remember that the collagen is required in the formation of bones. Now, if the collagen is weak, the bone is soft. So you have a very soft bones. The bones is very malleable. Okay, so hydrogen bonding, hydrophobic interactions. No? This can influence how the protein will be formed. Okay. Peptides are polyelectrolytes. Okay. Polyelectrolytes. What do you mean by that? What is an electrolyte? What are electrolytes?
Ions in the body.
Ions.
What do you put into the battery of your car?
Ion.
Ions.
Electrolytes. That's electrolyte. No? Okay. So polyelectrolytes is because of the ability of the R group, and also the peptide bond, to have a positive or negative charges. So it becomes a polyelectrolyte, whether the R group is acidic or basic. And in the string of amino acid, there are amino acids that are basic and amino acids that are acidic. So it becomes a polyelectrolyte. Thank you for attending. Are you all present?